Welcome to Edstrong Learning. Do subscribe to the channel if you haven't yet. In the previous video, we started discussing about the human cell. Cell and its structure, cell components have been discussed in earlier video. In this video, we will be looking at genetics of a cell and some important types of genetic diseases. The first topic of today's video is genetics. What is a gene? A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. The Human Genome Project estimated that humans have between 20,000 and 25,000 genes. Protein synthesis. The gene consists all the information required for the protein synthesis. This information is stored in DNA of a cell. DNA, with the help of RNA, ribonucleic acid, completes the process of protein synthesis. RNA, ribonucleic acid, is a molecule essential in various biological roles in coding, decoding, regulation and expression of genes. It is single-stranded. There are three types of RNA: 1. mRNA, messenger RNA; 2. tRNA, transfer RNA; 3. rRNA, ribosomal RNA. Transcriptome is the sum of all the mRNA molecules, expressed from the genes of an organism. The protein synthesis takes place in two stages. 1. Transcription. mRNA carries complementary genetic code copied from DNA. This complementary code acts as a command to which type of protein is to be synthesized. 2. Translation. rRNAs combine with protein from food and enzymes in the cytoplasm to form ribosomes, which act as the site of protein synthesis. These complex structures travel along the mRNA molecule during translation and facilitate the assembly of amino acids to form a polypeptide chain. They interact with tRNA and other molecules that are crucial to protein synthesis. tRNA are an essential component of translation, where their main function is the transfer of amino acids during protein synthesis. There are two types of cells in the human body: 1. Somatic cells; 2. Germ cells. Somatic cells are cells whose genetic material cannot be passed on to future generations of people. The vast majority of cells in the human body are somatic cells. Examples include the tissues that make up our skin, muscles, lungs, liver, and heart, as well as blood cells. Germline cells, or germ cells, are cells whose genetic information may be passed down to future generations of people if they resulted in a pregnancy that came to term. Human germline cells are sperm or egg cells, embryos, or reproductive stem cells. How do cells divide? There are two types of cell division: mitosis and meiosis. Mitosis is a fundamental process for life. During mitosis, a cell duplicates all of its contents, including its chromosomes, and splits to form two identical daughter cells. Because this process is so critical, the steps of mitosis are carefully controlled by a number of genes. When mitosis is not regulated correctly, health problems such as cancer can result. Meiosis ensures that humans have the same number of chromosomes in each generation. It is a two-step process that reduces the chromosome number by half, from 46 to 23, to form sperm and egg cells. When the sperm and egg cells unite at conception, each contributes 23 chromosomes, so the resulting embryo will have the usual 46. Meiosis also allows genetic variation through a process of DNA shuffling while the cells are dividing. The mitosis and meiosis happening in giving birth to a new child is shown here in a tree diagram. After an egg is formed, the 46 chromosomes undergo the process of mitosis to form an embryo, and on further mitosis, a baby is developed. How is sex determined? A human being has 23 pair of chromosomes, of which 22 pairs of chromosomes are same in both male and female. These similar 22 pairs are known as autosomes. The 23rd pair is responsible for the determination of sex of an individual. The XY pair indicates male. The XX pair indicates female. As the 23rd pair is responsible for the determination of sex of an individual, they are known as sex chromosomes. Now let's see how a child is formed on chromosomal basis. Follow the yellow lines in the tree diagram to know boy child, the red lines for girl child. If one can notice in this diagram that only male parent is responsible for the gender of a newborn baby. Some special cases when more than one child is conceived in the same pregnancy. Semi-identical twins happens in very rare cases. Due to the fusion of 1/2 sperm, they are 75% to 90% similar. Each of the semi-identical twin can be of either gender. Identical twins happens due to fusion of 1/1 sperm. The nucleus duplicates itself in the first cell, leading to the formation of two nuclei with same characters. They are 100% genetically similar. Only difference is in their fingerprints. Both of the identical twins would be of the same gender. Non-identical twins, due to the fusion of 2/2 sperm. This is due to the fact that in some cases women produce 2 ova at the same time. Very rare case. Each of the non-identical twins can be of either gender. In the first table, some human characteristics are listed along with their dominant and recessive traits. The second table explains about the child characteristics, depending on the parent characteristics. For example, the male has dominant trait of curly hair, and the female has a recessive trait of straight hair. The child is born with the dominant trait of curly hair. Similarly, other characteristics. What is mutation? A gene mutation is a permanent alteration in the DNA sequence that makes up a gene, such that the sequence differs from what is found in most people. Mutations range in size. They can affect anywhere from a single DNA building block, base pair, to a large segment of a chromosome that includes multiple genes. These changes can be caused by environmental factors such as ultraviolet radiation from the sun, or can occur if an error is made as DNA copies itself during cell division. Today's Earth biodiversity is a resultant of various mutations over time. Now, let us look into the second topic of our video, genetic diseases. The first genetic disease is X-linked inheritance. It is a sex-inherited/sex-linked disease. It is due to the X chromosome present in the sex chromosome. Hemophilia disease results in no blood clot. Color blindness due to X-linked inheritance results in non-identification of red, green mostly. The X chromosome is significantly longer than the Y chromosome, and contains hundreds more genes. Because the additional genes in the X chromosome have no counterpart in the Y chromosome, the X genes are dominant. For example, when a healthy female and a hemophilia male have a child, the boy child will be healthy and the female child will also be healthy but a carrier of that disease for next generations. The second genetic disease is Y-linked inheritance. It is a sex-inherited/sex-linked disease. It is due to the Y chromosome present in sex chromosome. It is seen only in male. Hypertrichosis and pattern baldness are two major disorders in this inheritance. The third disorder is Down syndrome. It is caused as the 21st pair is trisomy, set of 3 chromosomes. The characteristics of this syndrome are slow learning capacity, slow growth, short height, heart defects, early deaths. This syndrome can occur in both male and female. The fourth genetic disease is Turner syndrome. It results when one of the X chromosomes, sex chromosomes, is missing or partially missing. 44 plus X missing. Only in female. Short stature, infertility, heart defects and certain learning disabilities. The fifth disorder is Klinefelter syndrome. An extra copy of X chromosome, 44 plus XXY, in male. Gynecomastia, low testosterone level, reduced muscle mass, no facial or body hair, mentally disturbed and low learning capacity. The sixth genetic disease is super males. Due to an extra copy of Y chromosome, 44 plus XYY, in male. Tall, highly aggressive, high sexual desire, learning problem. The seventh disorder is albinism. Due to a defect in gene, both in male and female, the affected gene doesn't produce melanin. Melanin gives dark color to skin, pale skin, eye conditions and are sensitive to the sun. The eighth genetic disease is sickle cell anemia. It is a group of disorders that affects hemoglobin, the molecule in red blood cells that delivers oxygen to cells throughout the body. Sickle cell disease is caused by a mutation in the hemoglobin beta gene found on chromosome 11. Symptoms: feeling fatigue, pale yellow skin, joints and body pains, anemia, organ failure in critical conditions. Treatment depends on severity. Treatments include medication, blood transfusions and rarely a bone marrow transplant. In sickle cell anemia, the biconcave RBC cells are converted into sickle-shaped RBC. The ninth genetic disease is thalassemia, a blood disorder involving lower than normal amounts of an oxygen-carrying protein, that is deficiency in amounts of hemoglobin in blood. Symptoms include fatigue, weakness, paleness and slow growth. Mild forms may not need treatment. Severe forms may require blood transfusions or a donor stem cell transplant. The 10th genetic disease Credushat, cat's cry, syndrome. Also known as 5p- syndrome. It is a chromosomal condition that results when a piece of chromosome 5 is missing. Infants with this condition often have a high-pitched cry that sounds like that of a cat. In this video, we have completed the genetics, protein synthesis, cell division, genetic diseases. In the next video, we will be covering biotechnology and gene therapy. Thank you for learning. If you liked the video, make sure to hit the like button, and be sure to subscribe, to never miss an update from Edstrong Learning.